So last time, before the phone rang, we left off with our discussion of what goes on in the renal capsule. And we were talking about the epithelium that is going to cover the glomerula and the glomerulus, the blood vessels within the glomerulus, and these visceral epithelium is going to be consistent of the podocytes, these big cells with these feet-like structures that make our filtration slits. Now let's have a look at it. So here we have the renal corpuscle right here. Here is our glomerulus, here are our podocytes. Here you can see the pedicels, or the foot-like structures that create this filtration slit here. And so all of this underneath, this red stuff, is part of the glomerular capillary. Now we're going to have our capsular epithelium, which is the simple squamous epithelium here. And notice that it is continuous with the visceral epithelium, which are made up of these podocytes surrounding the glomerular capillaries. Here we have the capsular space. And this is where the filtrate is going to go. So the filtrate will be passed through the three-layered membrane that we talked about earlier. So here we're going to have our afferent arteriole bringing in our blood. And you'll notice there's some smooth muscle around here that can help control the diameter of this. And that's going to enter into the glomerulus. And then we're going to have the process of filtration happening within the glomerulus that's going to be due to the hydrostatic pressure of blood pressing up against these walls. And of course, just like any filter system, if you have enough pressure exerted on the filter, fluid will move through the filter, and then the solutes that are too large to pass will stay behind. And those will then be taken along with the blood out of the efferent arteriole, which will then go and feed into the peritubular capillaries. But now let's have a closer look right here. So when we think about blood hydrostatic pressure, we're thinking about the pressure that the blood or the fluid is exerting on these walls, which in this case is going to be a filtration system comprised of our endothelial fenestrated blood vessels, our dense layer, and our filtration slits made up by these podocytes. So just the pressure of fluid moving through here is going to contribute to our blood hydrostatic pressure. And anything that can move through this filtration membrane will. So that will be blood plasma and very small solutes. Other things will stay behind. Now there are two major obvious ways that we could regulate the blood hydrostatic pressure. If we increase the diameter of the afferent arteriole, more blood can come in. And so more blood is going to mean higher pressures. So that would force more solute, or I should say more filtrate, through the filtration membrane. And whatever solute is small enough to pass through the filtration membrane will also end up in the filtrate. Now, if we decrease the diameter of the efferent arteriole, then we would actually restrict how much blood could leave here. So it'll back up in here, so we produce more pressure. So that there again, that would lead to a higher filtration rate. And the rate at which we can filter fluids through the glomerulus is called the glomerular filtration rate. And we'll talk a little bit about that later on. But suffice it to say, we've got these two ways of controlling our blood hydrostatic pressure. We can dilate the afferent arteriole to increase pressure. We could constrict the efferent arteriole, again to increase pressure, because blood would back up in here and it would take longer to be able to get out the efferent arteriole, and there would be more peripheral resistance of these vessels. Now also consider for a second, if you constricted the afferent arteriole, then you'd be kind of turning down the blood flow into this area, and so there would not be as much pressure. So you can reduce pressure at the glomerulus by restricting the diameter of the afferent arteriole and not letting as much blood in here. Likewise, if you increase the diameter of the efferent arteriole, blood could exit faster. And so you would actually have, again, a decreased pressure. And we will see that we will have, again, our blood hydrostatic pressure is going to come into play here. That will oppose capsular hydrostatic pressure, because we will have some fluid in the capsule, and it's going to take a while in order to get through the proximal convoluted tubuli. There will be resistance provided by the tubule. So the filtrate will have its own hydrostatic pressure, but it will not be as great as the hydrostatic pressure within the glomerulus. We're also going to have our blood colloid osmotic pressure. And so as we know, we've got albumins and other things in here, and large solutes. And remember, that which does not pass through the filtration membrane is going to be left behind. and remain within the blood plasma, and will not be filtered. So we're going to have a higher blood colloid osmotic pressure within the glomerulus than in the capsule. So we will have things like glucose, a few fatty acids, some very small albumins and the like in here, some ions. But the main thing to consider is that the large plasma proteins that are left behind within the glomerulus are going to be contributing the most to the blood colloid osmotic pressure. And since what we have on this side are smaller solutes, amino acids, fatty acids, glucose and the like, and some very small ions, the capsular colloid osmotic pressure will be very, very low by comparison to the glomerular, or what I should say, blood colloid osmotic pressure, which is contained within the blood within the glomerulus. So that basically is going to mean that the capsular colloid osmotic pressure will be virtually zero. So unless something happens and there's damage to the glomerulus and to this filtration system and albumins are getting out, then we can consider the capsular colloid osmotic pressure to be zero. And that's going to be important because as we will see that the two hydrostatic pressures within the glomerulus and within the capsule, as well as the blood colloid osmotic pressure, are going to be the main drivers of this filtration process. Remember, filtration is passive. It's simply driven by our hydrostatic pressures and to a lesser extent our blood colloid osmotic pressure. And we will see that the filtration is not only passive, but once again it happens only in the glomerulus. So down here we've already produced our filtrate. So our proximal convoluted tubuli will be where we're going to start to pick up important things that were filtered out that we really want to hold on to. So we will reabsorb things like glucose and ions that we need and fatty acids here, whereas the glomerulus is such a good system that a lot of things will be filtered out, and we'll need to recapture some of those things. All right, now let's look over here. This is going to be our juxtaglomerular complex. This, as we've said before, is our distal convoluted tubuli, and it will come and make sort of an interface here between our afferent and efferent arteriole. And then we're going to have, within this juxtaglomerular complex, we're going to have regulation of some of the things that are going to go on within the glomerulus. So let's have a look. We've got fenestrated capillaries within the glomerulus. So basically they've got little windows in them. They've got much larger openings than a regular blood endothelium. So we're going to have some supporting cells called mesangial cells that are going to be between adjacent capillaries. And these are called intraglomerular mesangial cells, and they will control blood vessel diameter within the glomerulus. So here we have a cross-section through part of some blood vessels, the dense layer, and some podocytes around this. So this is part of our glomerular capillary system. Here is our capsular epithelium, and here's our capsular space. So you can see within the blood vessel we would have red blood cells and the like. So one of the things you will notice is that we have these mesangial cells, and these are called intraglomerular mesangial cells because if we go back here, there are going to be some extraglomerular mesangial cells that reside within the juxtaglomerular complex. Now let's have a closer look once again. So our filtration membrane is going to be made up of our fenestrated epithelium, which in blood vessels is called endothelium, our dense layer here, and our podocytes that make these filtration slits. And so it's a progressively selective membrane. So as we go through each of the layers, smaller and smaller solutes will be filtered out. And so we will have only a very pure, much purer filtrate on this side than we have inside the blood vessels. All right, so we've talked about these three things. We've seen them now. We know that we have passive filtration as the solutes are going to enter the capsular space. We're going to have some metabolic waste, some excess ions. We're also going to have things we want to hold on to. A little bit of glucose, free fatty acids, amino acids, vitamins, and the like. But pretty much we're going to have a protein-free filtrate. So all of our albumins, with the exception of very small ones, will be left within the glomerulus. But when this filtrate that was produced within the renal corpuscle, when it moves into the proximal convoluted tubuli, this is where we're going to start our resorption of material. So this is where we're going to pick up our useful things like our glucose, fatty acids, amino acids, and so forth, so it does not leave with urine, because these are valuable things and we would really have to eat a lot, do a lot more, spend a lot more energy getting these things again from the outside, when it's much easier just to reclaim them before they leave in the urine. So we get a lot of resorption that's going to occur within the proximal convoluted tubuli. We will have some secretion within the proximal convoluted tubuli as well, but as we will see, the renal tubuli will change in terms of what goes on in it as we move through different parts of it. So the loop of Henle will have something a little bit different going on. We'll have, in the descending limb, a lot of water osmosis out of the filtrate back into the interstitial fluids. On the ascending limb, we're going to have a lot of recapturing of ions like sodium and chloride back into the interstitial fluids. And then when we get into the distal convoluted tubuli, we'll have some resorption, but we'll have a lot more secretion of things. And there's a lot of variable secretion depending, as we've seen earlier, on some of these hormonal controls. All right, so as we said, the proximal convoluted tubuli is what leaves that glomerular, well, that whole renal corpuscle. So it's going to take the result of the glomerular filtration and basically start the next batch of processes, starting with resorption of a lot of these materials. All right, the endothelial lining, or sorry, the epithelial lining, this is not a blood vessel, the epithelial lining of the proximal convoluted tubuli is simple cuboidal with lots of microvilli, as we've seen. All right, we're going to secrete some substances into the lumen, but we're going to have a lot of resorption going on here. All right, so we're going to have, we call these cells tubular cells because they reside within the kidney tubules. So, as we've said, they're going to absorb important things that we want to keep and release things that we don't want into the filtrate. It's going to absorb stuff we want to keep and release those things into the peritubular fluid. That is the interstitial fluid, which will then, that fluid will be picked back up by capillaries that are passing through it. So we'll have an exchange between the blood capillaries and the interstitial fluid as well. So basically, the tubular cells are going to absorb things we want to keep, pass that into the interstitial fluid, and then that will then be returned to blood circulation. And we will also have some secretion from the tubular cells. And right now we're going to concentrate on the proximal convoluted tubuli. So we will have some secretion of materials into the lumen of the tubule, and those are the things that will leave with the urine. All right, then we descend into the nephron loop, or loop of Henle, and this is what's going to turn towards the renal medulla. In those juxtamedullary neurons, this is the nephrons, it's going to dip way down into that renal medulla. So the descending limb is where we're going to have the tubular fluid, the filtrate, flowing towards the renal pelvis, but then of course it will climb back up. The ascending limb is where it goes back up towards the renal cortex, and we've got thick segments and thin segments in each limb. The thick segments have a cuboidal type of epithelium, whereas the thin segments go back to that simple squamous epithelium. And so the descending limb is going to be somewhat similar to the functions of the proximal convoluted tubuli. This really pertains more to the thick ascending limb, which we'll talk about a little bit later, where we're going to pump lots and lots of sodium chloride out of the tubular fluid into the interstitial spaces. So the thick ascending limb. So on the ascending limb, especially the juxtamedullary neurons, we're going to create a very high solute concentrations in the peritubular fluid. That is the interstitial fluid that surrounds these things. So when we have sodium and chloride in here, you can imagine that's going to make the peritubular fluid very salty. And that is going to increase osmosis of water out of the descending limb into the peritubular fluid. All right, so as we've said, this is what's going on in the thin segments. Freely permeable to water, but not so much to solutes. Solutes are going to stay behind. But the water is going to be osmosing, if that's a verb, out of the tubule into the peritubular fluid. So this is going to concentrate the filtrate within the tubular fluid to be less water, more solute. In the thick ascending limb, then we are going to, this is where we're going to have a lot of sodium and chloride movement out of the tubular fluid into the peritubular space, or the peritubular fluid. This is also at the very end of this thing, we're going to have the distal convoluted tubuli. And once again, here's our picture for reference. Here's our renal corpuscle. Here's our proximal convoluted tubule. Here's our descending limb of the loop of Henle. And this is the thick portion. Here's the thin portion. Lots of water moving out here. Then we're going to have the thick limb, the ascending limb, the TAL, or the thick ascending limb, sometimes just simply referred to as TAL, of the loop of Henle. And this is where we're going to resorb some sodium and chloride, a lot of it. And then we're going to get to the proximal convoluted tubuli. Once again, distal convoluted tubuli, sorry. Once again, the distal convoluted tubuli, where we're going to have the selective resorption and secretion of certain solutes and the selective resorption of water, depending on how much blood plasma volume we have. All right, so just to recap, renal corpuscle, proximal convoluted tubuli, so named because it's closest to the renal corpuscle. Distal, because while it's in physical proximity, it's still the farthest in terms of the distance traveled along the nephron loop. So it's the distal end of the nephron, the whole renal tubule. So the renal tubule is this whole thing. So the proximal convoluted tubuli, the loop of Henle, and the distal convoluted tubuli, the latter of which is so named because it's the last section in this tubule. All right, so the distal convoluted tubuli is that third segment, and as we know, the initial portion is what's going to pass between the afferent and efferent arterioles, and it's going to have a smaller diameter than the proximal convoluted tubuli, and as we saw earlier, the epithelial cells are going to have very few microvilli. And here we're going to have a lot of active secretion of ions, acids, hydrogen ion, we're going to see a lot of that, some bicarbonate ion, drugs, inactivated drugs, and toxins. We're going to have selective resorption of sodium ions. Remember, our aldosterone will increase this process of sodium, reclaiming sodium ions. And calcium as well, so we're going to have some calcium ions that will be resorbed from the tubular fluid. And we will also have selective reabsorption of water that will help concentrate the tubular fluid as it goes into that collecting duct. And so by the time we get into the collecting duct, most of the urine will have its constituent parts by this point in time. So this is almost the end stage of the creation of urine. I mean, it'll still pass through the collecting duct. There'll be a little bit more resorption maybe. There are some aquaporins in that region that can be under the effect of antidiuretic hormone. We'll also see that, if you recall, calcitriol is going to increase our propensity to absorb ions, calcium ions, in digestion. It will also increase the amount of calcium ions we resorb in the tubular fluid so that we retain this calcium instead of excreting it away. And so that will have the effect of bringing up our blood levels of calcium. All right, so our juxtaglomerular complex. This is an endocrine structure. It's going to be what's going to secrete our erythropoietin. That's going to increase our red blood cell production and thicken up our blood. So we're going to have more formed elements, and of course that's going to make our blood more viscous. It's going to increase blood pressure. It's also going to increase the enzyme renin, which is a key component of the renin-angiotensin-aldosterone system. And what is the purpose of that? Is to maintain enough blood pressure to perfuse the glomeruli and have enough hydrostatic pressure to cause filtration. So the juxtaglomerular complex will be formed by a very sensitive set of cells within the distal convoluted tubuli that are the macula densa, and these will be in contact with the juxtaglomerular cells. And these things, and some of these are the external extraglomerular mesangial cells. And this will be what creates this juxtaglomerular complex. All right, so our macula densa, as we know, are epithelial cells of the distal convoluted tubuli. This is going to be right near that renal corpuscle. And these are basically really tall cells. They're going to have some densely clustered nuclei. We'll look at it in just a second. And then we have our juxtaglomerular cells that are sort of wedged between the macula densa, the afferent and efferent arterioles. And this is going to be monitoring what's going on within the macula densa. All right, so here it is. Here's the macula densa. Here are our juxtaglomerular cells, which include some extraglomerular mesangial cells. And these things together are going to create what is called the juxtaglomerular complex. All right, so when we look at, now that we've looked at the entire nephron, the nephron is going to empty into the collecting system. So that's where the distal convoluted tubule basically dumps into the collecting duct. And then we're going to have several collecting ducts that are going to converge into a papillary duct. The papillary duct will then empty into a minor calyx, which then empties into a major calyx, then empties into the renal pelvis, and then down the ureter towards the bladder. All right, so basically the collecting duct, there's going to be a little tiny bit more adjustment of the composition of the urine. So this is going to determine the final osmotic concentration and volume of the urine. So what is the goal of urine? Well, one, to get rid of stuff, but also to maintain homeostasis. And a big part of that is regulating the blood volume and concentration, composition of blood, concentration of certain ions and solutes within the blood. So we got to get rid of metabolic wastes. If we don't get rid of them, they can be toxic and they can eventually kill us. If you have high creatinine levels or these kinds of things, that's bad. So we're going to get rid of metabolic waste products through urine. It's going to adjust the concentrations of ions like sodium, potassium, calcium, and so forth within blood plasma. And it's also going to regulate the amount of blood plasma and the volume of it by how much fluid we retain or how much we excrete. All right, so we have 3 organic waste products that we're going to look at: urea, creatinine, and uric acid. And creatinine is going to be a breakdown of creatine phosphate, which is a muscle product. Urea is going to be the coming from the deamination of amino acids, and uric acid is going to be the breakdown of RNA. So all of these organic wastes, which are dissolved in the bloodstream, will be eliminated through urine. And of course, since they have to be dissolved in something, they have to be dissolved in some kind of fluid, water. And so that's why they can only be lost through the urine. So these are eliminated while dissolved in urine. All right, so the kidneys are producing very concentrated urine. So 1,200 to 1,400 milliosmoles per liter. So 4 times the plasma concentration. But we're going to retain a lot of the water that we originally filter. So a lot of the water that passes through the glomerulus, a lot of it will be reclaimed within the tubule. And if that were not the case, we would dehydrate very, very quickly. So as we know, we've got the kidney is not only going to filter the blood plasma, but we are going to concentrate that filtrate, reclaiming a lot of that water. Otherwise, you'd be dead very soon. And we're also going to retain the materials that we want to keep, like sugars and amino acids. All right, so when we look at urine formation, we've got filtration, which we've already talked about, happens only at the glomerulus, at the renal corpuscle. So the glomerulus does the job of filtration. As I say, it's a good test question. Resorption is going to happen all along the kidney tubule. It's going to vary by section, so we're going to have a lot more resorption in the proximal convoluted tubule than in the distal. We're going to have a lot of secretion all over the place, but most specifically at the distal end. We're going to have a lot more secretion at the distal end, the distal convoluted tubule. And what we're going to do is we're going to see that we're going to have a lot of water and solute resorption very much toward that proximal convoluted tubule, as we've said. And active secretion all over the place, but we're going to see more active secretion at the distal convoluted tubule, quite a bit at the proximal one, but more, nevertheless, a bit more at the distal end. And then the long loops of the juxtamedullary neurons are going to be very important for concentrating that urine, concentrating the solute, resorbing a lot of water. And as you can already sort of see where this is leading, we've got a very high salt solution in the interstitial fluid through which the tubule, the loop of Henle, is running. So obviously, if you think we've got very high amounts of sodium chloride in the interstitial fluid, which is outside the kidney tubule, we're going to obviously induce water to move toward the solute, so out of the tubule into the solute. And we're going to concentrate a lot of that sodium chloride in that thick ascending limb of the nephron loop. So this is where a lot of our sodium chloride will be pumped out, whereas the descending limb will be completely impervious or impermeable. The thin descending limb will be impermeable to those solutes, those ions. All right, so just to recap, here's our renal corpuscle, here's our glomerulus. Usually it'd be covered with podocytes, but they didn't show that here. We're going to have our filtrate, which is then going to go into the proximal convoluted tubule. We'll have a lot of resorption of water here. We'll have a lot of resorption of other solutes, amino acids, sugars, and so forth. We can have some secretion into the proximal convoluted tubule of different waste products. And then we're going to enter into the thin descending limb of the nephron loop, where we're going to have a lot of water resorption going on here as well. And then by the time we get to the thick ascending limb of the nephron loop, we're going to be picking up especially things like sodium chloride. That's going to concentrate this surrounding interstitial fluid with salt. What's that going to do? That's going to pull more water out of the nephron loop. And we'll see a little bit later, this is part of a process we call countercurrent multiplication. And we're going to look at that in a lot of detail later on. But for now, suffice it to say, we get a lot of osmosis of water because this is freely permeable to water and pretty impermeable to other solutes. Whereas we get up here, this is impermeable to water, but we're going to have active removal of sodium chloride through this thick ascending limb. And then we're going to have variable resorption and secretion of things along the distal convoluted tubule, which then leads into the collecting duct, which will have our final adjustments on the concentration of urine, of what solutes are in it, and what the volume of it is. And then by the time we get to the papillary duct, urine is in its final form, and it will then move on through those calyces into the renal pelvis, and then through the ureter toward the urinary bladder. All right, so here's a nice little table. And these are normal laboratory values for the solutes that you would find in plasma versus urine. And so you can see plasma concentrations of sodium, of potassium, chloride, bicarbonate ion, and how they would vary in blood plasma versus in urine. And so if we look in plasma at glucose, for example, we have high glucose levels in plasma as compared to the urine. So we've recaptured almost all of the glucose that was filtered out at the glomerulus. So lipids, once again, lipid concentration in plasma is quite different from lipid concentration in urine. Amino acids, we get quite a bit more amino acids in plasma than in urine. So most of these valuable things have been recaptured by the kidney tubule or the renal tubule. One thing to note, proteins. You should not have any proteins within the urine. If you have proteins within the urine, then this is a sign of some kind of serious metabolic disorder or problem. Right, we have urea, creatinine, and uric acid, as well as pneumonia. We see the levels in plasma are low compared to in urine. So you can see that urea is doing its job. Urine is doing its job by excreting, or basically carrying the solutes, these toxic solutes, out of the body, like urea, creatinine, and uric acid, as well as some ammonia. All right, so filtration. This is where everything comes together. This is the hydrostatic pressure on those walls of the glomerulus that's going to force water through the membrane pores. Small solutes can also get through, and everything else stays behind. And so, as we've seen with other capillaries, we've studied this at the capillary beds, we've studied blood hydrostatic pressure versus colloid osmotic pressure. And we've talked a bit about the different types of endothelial cells. And we've talked about fenestrated endothelial cells, and we talked about ones with sinusoids. But now we're going to focus on the fenestrated ones, because we are going to have these special membranes that are going to restrict all of our circulating proteins. So we're going to have all kinds of processes going on in the kidney: diffusion, osmosis. We're going to have some channel-mediated diffusion, in other words, carrier-mediated diffusion, and we're going to have some carrier-mediated transport as well, so both passive and active. So channel-mediated diffusion is going to be basically passive. Remember our aquaporins, this is where water is going to go through. We're going to have other channels that will be able to pick up different solutes. Carrier-mediated transport, we're going to have, as we've seen before, with like glucose transport and these kinds of things, like little doors that open so that the molecule fits in. The door closes on one side, opens on the other, and dumps the molecule into the other side of the wall, or the membrane. All right, so we're going to have facilitated diffusion. This is where, say, here's your transport, and you've got more concentration up here than you do in here. So your molecule comes into your transporter, and then the transporter closes, opens it to the other side, and dumps the solute into the area where there's less of it. So this is all going to be basically diffusion, going down its concentration gradient. Active transport is where we are transporting these molecules against their concentration gradient. Co-transport is where we might be using, for example, in the sodium-potassium pump is a good example that we've seen before. So, that's countertransport, sorry. Anyway, we're going to have co-transport, which is going to transport two things at once. So a good example of this is the sodium glucose transporter that we've seen in previous chapters. And then we'll have countertransport, and that's like the sodium potassium pump, where we've got 2 potassium coming in and 3 sodiums being kicked out, so of a cell. So these are our types of carrier-mediated diffusion. We'll see every one of these within our renal tubule. All right, so we've talked about all of these. I just went through most of them. With any carrier-mediated transport, remember, they are like enzymes. So we can only, let's say we've got a revolving door. We can only open it so many times a minute and let so many molecules in. And once that thing is working at full capacity, it is saturated. We can't go any faster. So we can actually saturate some of these transporters. And this is exactly what happens in diabetes. We have so much glucose in blood plasma that our glucose transporters to resorb that glucose get saturated. And we can't make them work any faster. And so a lot of that glucose gets excreted in urine. All right, so we've got basically a transport maximum. And so this is what that speaks to. If the nutrients concentrations rise in the tubular fluid, the reabsorption rate will increase until the carrier proteins are saturated. And so if concentrations are higher than maximum transport, obviously some will be left within the tubular fluid and will appear in urine. And so this is why tests for glucose in diabetes can tell you if you've got glucose in urine, that can be an indicator of diabetes. All right, so we have a renal threshold, and this is the plasma concentration at which a specific compound or ion will begin to appear in the urine. So that's going to vary with the substance that we're looking at.